DNA sequencing that was developed in the late 1970s. Principles for DNA sequencing methods are to separate piece of DNA of different sized, all starting from the same point and ending at different points. Second principle is the separation of these different sized pieces of DNA by gel electrophoresis. Number third is reading of sequence from the gel. For DNA sequencing, two methods were developed, that are Sanger method and Maxam-Gilbert method. Sanger method was developed by Friedrich Sanger and Andrew Coulson in 1977, while the Maxam-Gilbert method was developed by Alan Maxam and Walter Gilbert in 1976 to 77. Here we will discuss the Sanger method. So the Sanger method, that is known as Sanger sequencing. Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain-terminating dideoxynucleotides by DNA polymerase during in vitro DNA replication. And Sanger method was the most widely used sequencing method for approximately 25 years. So the standard method used for DNA sequencing is the Sanger's enzymatic method. If we look at the procedure of Sanger method, here double-stranded DNA is taken. This double-stranded DNA is denatured through sodium hydroxide to convert them into single-stranded DNAs. From these single-stranded DNAs, one strand is used as a template strand, and this strand is tagged at 3' end. Look here, that is the template strand, which is a single-stranded DNA with unknown sequence. Blue means that it is tagged with blue dye. Then the radioactively labeled primer is used here. Primer is a short sequence from 5 to 3' direction which have free hydroxyl group at carbon number 3rd to attach further nucleotides. Then this template strand is divided into 4 tubes that are labeled with guanine, adenine, thymine and cytosine tubes. Here the reaction mixture is added that is consists on DNTPs, DNA polymerase and the template DNA. And the terminators that are dideoxy GTP is added to the G tube, Dideoxy ATP is added to the A tube and dideoxy TTP is added to the T tube, while dideoxy CTP is added to the C tube. Dideoxy means that commonly and the commonly and the DNTP is where one oxygen is missing at carbon number second in the pentose sugar, so they are DNTPs, while in DDNTPs there is another oxygen missing at carbon number third as well. So the carbon number third, which has free hydroxyl group where the further nucleotides are attached, so due to the missing of oxygen at carbon number third, no further nucleotides will be attached here, so the dideoxy NTPs are used as terminators in this procedure. So here in all the four tubes DDNTPs are added. After addition of the reaction mixture as well as all these DDNTPs, these four tubes are placed into the PCR machine where multiple copies of these DNA are formed, means DNA synthesis will takes place in the PCR machine. After DNA synthesis, fragments of DNA are separated through a process known as gel electrophoresis, and after that gel electrophoresis, the sequences are read out through autoradiography where the X-rays are used.